30 kb chromosomes like Tinker Toys. Now, the synthetic bat genome that we made using Blue Heron and BioBasic basically is interchangeable with the Urbani clone. The only real difference was that we broke the F fragment into 2 pieces so that we could play with the receptor binding domain easily if this thing didn't turn out to be infectious. And in fact, we made this clone, we built the full-length cDNA, we drove transcripts, electroporated that into cells, and we could see evidence of replication by the synthesis of subgenomic messenger RNA, but we couldn't culture the virus and we couldn't pass it from cell to cell. So clearly there was probably a defect in entry. To solve that problem, we used literature data that has suggested that RBD domains of coronaviruses may be interchangeable between species. So we took the human, the Urbani epidemic receptor binding domain, that's 210 amino acids in length, and dropped that into the bat genome backbone, producing a chimera with the receptor binding domain driven from the epidemic strains. Now, when we built that clone, drove transcripts and electroporated that into cells, we got a virus that could replicate quite efficiently. This is just some growth curve data showing, I think the black box is the Urbani wild type and the white circles or open symbols are the bat viruses. You can see they replicate exactly as good as the Urbani epidemic strain at low and high multiplicities of infection. They recognize the human ACE2 receptor in DBT cells at low and high multiplicity infections and grow just like the epidemic strains. And they also retain the ability to use the civet ACE2 receptor just like the epidemic strains at low and high multiplicity. They also are capable of infecting human airway epithelial cultures and targeting ciliated cells just like the epidemic strain, and they grow to similar titer.